Hello, everyone, and welcome to phyloseminar.org. This is the first talk in a series of 3 talks on the Markov genealogy process. And the Markov genealogy process is definitely a brainchild of Aaron King and his collaborators, and so it's really exciting to have this series of 3 talks. And I'd like to acknowledge Tanya Stadler, who suggested that we have this session. Please use the YouTube live comment box to ask questions. And as I said, today's speaker is Aaron King. Aaron is the Nelson G. Hairston Professor of Ecology, Evolutionary Biology, and Complex Systems at the University of Michigan and an external professor at the Santa Fe Institute. He's an applied mathematician and a theoretical biologist whose work focuses on the dynamics of host-pathogen interactions from the scale of infections to that of epidemics. He's best known for his work developing computational methods for efficiently extracting information from noisy, incomplete observations and exploiting these techniques to obtain insights into the determinants of transmission and susceptibility on the basis of time series data of various kinds. Welcome, Aaron, and thank you for participating.
Thank you so much, Eric, for the invitation and for the opportunity to talk to this group. I'm really looking forward to it because I think this group will have a lot to say, and your suggestions, questions, and comments would be really valuable to me. And I hope maybe this stuff will ultimately be of value to you as well. So let me go ahead and get started. I guess we checked. You can see the cursor.
Yeah, looking great.
Okay, fantastic. So to begin with, because I don't want to forget to acknowledge a whole bunch of people that have been really important in the work that I'm going to talk about. Obviously, Kyung Lin and Ed Ionides, my collaborators, who very much played an important role in developing the theory that I'm going to talk about. But a lot of important conversations with people like Eric Foltz, Chris Moore, George Cantwell, Ethan Romero-Severson, Simon Frost, as we're instrumental in the developments that I'm going to talk about. And then there's been some funding as well to help keep the wolves from the door. So I'm going to, fundamentally, this is a fairly theoretical talk and a fairly mathematical subject. So I want to start by kind of trying to motivate it in terms of a problem that maybe would be familiar to many of us. And that's the problem of surveying for new variants of an infectious disease. And so SARS-CoV-2 variants, the top panel here, you've probably seen variants on this fairly frequently, but this shows the sort of frequency, the changing frequencies of different clades of SARS-CoV-2 virus over the period of time from 2020, early 2020 to late 2024. Early 2024, with the different groups shown in different colors, and this is the frequency ranging from 0 to 100%. And so one of the main tools that we have, of course, for doing this is based on genomic surveillance. So looking at, and in particular, reconstructed phylogenies based on those genome sequences. So, in the scheme of things, some of these variants take over quite quickly. Others fail to take over. Some show some interesting variation in their trajectories. And the question the broad question that I want to ask today is, how can we use information that is organized like this to infer things like what's going on in terms of strain variation in an infectious disease. Here's just another view of some of those same data. So let's just think for a moment, put ourselves in the position of trying to, looking at a few samples from a new variant and asking ourselves the question, is this variant on track to become the new dominant variant, supplanting all the existing ones, or is it not? Is it perhaps destined to be extinguished before it gets to be at appreciable frequencies? So that's an example of the kind of question, just an example, but it is an example of the kind of question that I think we're talking about today. So implicitly in that question, one probably has a model that might bear some resemblance to this. This is a compartmental model diagram of the kind that is commonly used in epidemiology. We have a pool of susceptible individuals who, when exposed to a virus, can develop a latent infection. Eventually, they become infectious. If they might recover and perhaps lose their immunity. But there's two pathways here representing perhaps two different strains of the virus. And so implicitly in the picture I was looking at before, there's questions about the relative rates of capture of new susceptibles by each of the strains, as well as important the important determinants in the form of rates of loss of immunity, durations of latent periods, and so on. So ultimately, a good way, or a way, certainly a reasonable and principled way to go about answering the question we were talking about, is to try to estimate the parameters of models like this, and perhaps given multiple models, different models that have different structures, to compare those models in terms of their ability to explain data. So we've known for a long time how to make inference on models like this, dynamical models that make predictions about the dynamics of a biological system. We've known for a long time how to infer the parameters of those models and to compare models on the basis of things like case report data, incidence data, seropositivity data, mortality data, time series data that directly or indirectly reveal the dynamics or reflect the dynamics of the system. What's been much less clear is how we go about using data like this. So how does one think about a phylogeny or a genealogy of a bunch of different pathogens in terms of what it has to tell us about the parameters of those models? So for example, just to kind of raise that question, right? If we think about this as a time series, right? Or if we think about this as revealing something that plays out over time, you can see that there are definitely periods of time when there were a lot of branching events, so presumably a lot of transmission, and other periods of time when maybe there was less. But the pattern of branching that we see does reflect to some extent transmission, but it also reflects the collection of samples, the surveillance effort itself. And so these things are mixed together in some way that is a little bit unclear. So, one way to think about it is to ask yourself, one way to put the question in perspective is to ask yourself, there was an ancestor to this clade of pathogens, of the samples here, and that was present, say, in the middle of 2022. Where was that? And how many were there? The fact that we see one of them, what does that tell us? Certainly it had to have been somewhere, but what are we to make of the fact that there wasn't very much of it? Another way of putting the problem is that we construct genealogies like this, looking backward in time, projecting from our samples backward in time to find the patterns of shared ancestry. But the kinds of models that we're interested in inferring can typically only be written and analyzed in forward time. So we have a kind of a fundamental mathematical problem here of how we put these things together. Oops. What did I just do? Okay. So just one more thing to notice here is so this is showing the actual numerical count of sequenced cases, and this is the frequency count that we looked at before. There's clearly variation in the invasion rates that we see, whether we look at it from either, whichever perspective we look at it from. And interestingly, there's also frequency dependence in this. So we've understood, we want to be able to estimate those kinds of things. And this is the subject of phylodynamics. So phylodynamics is a term that is used in actually have a wide array of meanings. And so I want to try to be very clear about what I mean by phylodynamics for the purposes of this talk. So broadly speaking, whenever we're talking about inferring something about the determinants of epidemic spread using genomic data, we can say we're doing phylodynamics. But here I want to focus much more precisely on using genomic data to infer the parameters of stochastic dynamic transmission models. And we can certainly talk as the talk goes on or afterwards about the relationship of this narrow definition to broader things. So there are many, basically the problem that we have is we have information in genome sequences and we want to get that to somehow make contact with stochastic transmission models. And there are multiple ways that we might go about doing that. One pathway that has been is fairly well trodden, and it's the pathway I'm going to focus on, is the pathway that lies through this tree-like structure of a genealogy. So I'm going to be talking about how we can get information to flow from genome sequences by way of a genealogy to stochastic transmission models. And of course, when we want to get information to flow that way, the most efficient way to construct a pipeline is to develop a generative model. So we can, in particular, I'm going to show you, and of course this is well known, but I'm going to talk quite a bit about how a stochastic transmission model induces a genealogy, how sort of genealogies are predicted by stochastic transmission models. And then it's a much more well-studied and subject of how the relationship between genealogies and genome sequences. And there are many different approaches that can be taken to that. So this is a relatively well-studied problem here. This is the one I'm going to be focusing on today. Let me make sure there's nothing more I wanted to say there. Okay. Now, of course, there are a number of problems with this, and I'm going to mention these problems because I don't want to talk about them. But again, at the end of the talk, we can spend some time thinking about these things if that's interesting. So the stochastic transmission model tells us something about the pattern of transmissions. And so this is a little cartoon that shows an infected individual who has, as this black line represents, a population of, say, viruses, pathogens inside that host. And at this point in time here, that population colonizes another host and founds another population in a different host. So that's a transmission event that can be represented in terms of a relationship among populations of pathogens. If we take a sample from each of these two individuals, and we can, through analysis of the similarities and differences among those sequences, genome sequences, we can try to infer something about the common ancestor and when that common ancestor was found. And of course, that will in general differ from this. Here I'm just representing in terms of a difference in timing. And of course, there are loads of reasons why this reconstruction itself can be difficult to do accurately. All of those things are important problems. I'm not trying to downplay them, but those are not the problems I'm going to focus on today. In fact, I'm not going to assume this, but I'm going to, what we can say is that it would be easiest to focus on what I am bringing that's new to the table if we just make the assumption, rather unreasonable, heroic assumption that we can somehow or other reconstruct the timing of this transmission event from those sequences in an accurate way. As I say, that's not an assumption, but for practical utility of these methods, that is an issue that has to be dealt with. Okay, so let's see. So as I say, if we take this point of view, we want to construct a pathway through the genealogy, then that actually factorizes the problem into two pieces. And here's a way of looking at that. Let's suppose we have a set of genome sequences. We have an inferred or perhaps a hypothetical genealogical tree that relates those sequences after this fashion. In addition, a model for the evolution of sequences, showing how sequences change along a lineage, along a tree. A dynamic stochastic transmission model, that's presumably the focus of our interest, as well as potentially other time series data. Then in order to infer the two model pieces, the central object of interest is the likelihood of those things, which of course is just the probability density. So this is just a generic probability density of observing the data given the model. And this, if you follow my reasoning so far, can be factorized into a term like this, or rather the integrand of this integral can be factorized. So for any given genealogy, the contribution to the likelihood is a product like this. And if this blue term here is known as the phylogenetic likelihood, and going back to Felsenstein and many people who've thought about that since, then maybe it makes sense to think of this red term as being a phylodynamic likelihood. Be that as it may, that's the element that I'm going to focus on. Okay, so I need to say a little bit about the existing approaches to this problem. And there are two large classes of approaches. The older, it goes back to Kingman's work on the coalescent processes, which essentially conceptualize, as you probably know, essentially conceptualize the coalescence of lineages as we go backward in time. Applied to the problem that we're studying here, one can justify this by making certain, in an asymptotic sense, by assuming the population size is large and the sample fraction is relatively small. Another interesting feature of this is that the samples themselves, the existence of the samples, has to be conditioned on, has to be taken for granted, and therefore it can contribute nothing to the existence of those samples contributes nothing. It's uninformative, right? So the fact that there were pathogens around to sample is itself not informed by the existence of samples in this approach. And all of these mathematical assumptions allow one to be able to relate the backward in time coalescent process with the forward in time dynamics. So all of that goes to say that if we have an arbitrary forward in time model or an inconvenient forward in time model, it can become tricky to relate the coalescent approaches formally to these things.
Maybe if you wouldn't mind just like
Yeah.
hopping back to the previous slide where you were talking about so let's see. So we're still obviously going to be conditioning on the set of genome. No, we're not. Let's see. Okay, I see.
No,
Okay,
so
so
yeah, so this is the likelihood of the genome sequences given the tree and
mhm.
whatever the sequence evolution model, substitution model is. And this is the likelihood of the tree together with what other data given the model,
Mhm.
right?
Okay.
So this is just an identity.
Right. I was just like thinking about, I mean, you were just setting up the fact that you're not going to be conditioning on having those exact genome sequences.
No, I'm
Right. So you're
well,
talking,
so right.
they're filling
So
on the left-hand side.
yeah, so I'm not conditioning that on the existence of the sequences. I am deriving the probability of having those sequences.
Great.
And
Thanks.
that can include the probability of being able to sample them at all.
Mhm. Perfect. Thanks.
Sure. So the second major category of approaches is really built up on the basis of the linear birth-death processes. And here, the tractability stems from the fact that in the self-similarity that comes with the independence of lineages in the linear birth-death process. So the ability to write down the likelihood here in general stems from that. Um, however, that linearity becomes problematic in the context, certain situations in the context of infectious disease propagation, because there are nonlinear phenomena that happen. So if we limit ourselves to certain portions of an outbreak or to right kinds of circumstances, then the linearity assumption becomes quite plausible. But there, of course, in general, we're interested in the nonlinear phenomena. It's also possible to view these as a basis for a more general approach where we linearize. And if we can make large population, small sample fraction assumptions, then we can get away with that. Of course, there are situations where we don't want to make large population size or small sample fraction assumptions for the simple reason that we have small populations and or we have very good dense sampling. More generally, the models that we're interested in as scientists, we choose for their scientific interest, for their ability to express our hypotheses about the way the system is working, not for their mathematical tractability. Or rather, it would be great, it would be nice if we could choose the models that we want to look at based strictly on their scientific interest and not on their their tractability. So what that boils down to in this context is that if I write down a stochastic process, in general, I can't reverse that. I can't reverse time in that in any meaningful computational sense. So that's a problem. So, more specific problems that emerge here is that, we may have additional time series beyond a genealogy. We typically will have things like case counts, mortality, and other kinds of epidemiological surveillance. Is it possible to simultaneously assimilate those data along with genomic data? The approaches that I talked about before, the standard approaches, typically rely upon fairly simple assumptions about the way the sampling is done. In the coalescent case, we condition on it. In other cases, we have to assume a fairly simple sampling model. But of course, our sampling effort and the mode of sampling that we have can vary importantly through time, and that should have an effect on things. How do we model that? I already mentioned getting rid of the large population, small sample size assumptions, and so there may be many ways to have all of these desiderata. But one way that's quite clear is if we could somehow figure out how to do simulation-based inference, because we can typically simulate any model that we might propose. If we could figure out how to do simulation-based inference, that would open the door to liberating us quite a lot. So that's what I'm gonna focus on. That's the goal that I've set myself, and I want to focus a little bit on some of the developments, the technical developments. So today, what I want to do is I want to show you how given any population process, any stochastic population process that induces a unique genealogy process, that is a Markovian process on the space of genealogies, how when we think about the relationship between that thing and observable data, we can basically view observable data as a pruned, obscured projection of these genealogies down onto the space of observable things. And what I'll claim today and show you in the next talk is that the exact likelihood of an observed genealogy satisfies a certain filtering equation that we can write down in general. As I say, in the next talk, which is in about a month, I'll go into more mathematical detail. All right, so a population process induces a process in the space of genealogies. What do I mean by a population process? So this is just a few examples of the kinds of models that one might want to be able to propose and be able to infer. So this is the one I showed you before, a two-strain competition model. Of course, we might want to complexify this, having more than two strains or having a more elaborate progression of infection. This is a simplified version. I'm going to use this as a workhorse for explaining what I'm going to do with the rest of the talk, just because it's the simplest. It's the simple version. It's the one strain version of this. But here's this is a kind of model where maybe we might apply this to SARS-CoV-2, at least in the early stages, where we have asymptomatic infections and symptomatic infections. We have different kinds of data, ancillary data from these two different things in the form of hospitalizations and deaths. But obviously, these are real infections and they affect the genealogies as well. How do we go about inferring the properties of these things? Here's a model of super spreading, behavioral heterogeneity. We have low spreaders and high spreaders and some behavioral switching between these things. But you can, for your own, I'm sure you can imagine like lots of different kinds of models. And so the class of models that we're going to be talking about today are basically discretely structured Markov processes. So that's what all these compartmental models are. Discretely structured means that they have a finite number of boxes or countable number of boxes, and otherwise are unstructured. And that's the category of population processes I want to think about. Okay, so we're familiar, let's just take this very simple one here. We're familiar with writing down things, how we can translate that diagram into a set of differential equations representing some kind of a deterministic dynamics that's associated with this kind of structure. What I want to point out is that there's other ways of representing this. So here's a Markov state transition diagram. So if we're at a particular state, the state variables in that model are just the compartment occupancies, S, E, I, and R, the numbers of susceptible, exposed, infected, and recovered individuals in the numbers in those four boxes there. And so if we're at a particular state, let's just call it X for that quadruple, then there's 5 different possible things according to that model that can happen at any given time. We can have a transmission event whereby the number of susceptibles goes down and the number of exposed goes up. We can have a disease progression event where an infected individual progresses to the infectious stage. We can have a recovery event, right? We can have a waning of immunity, or we can have a sampling occur. So this genomic sample is taken from that individual. So these are the 5 different basic, or the only 5 different state transitions that can happen. And I'll just claim, but that once we have a Markov state transition diagram, we can write down an equation that compactly encapsulates that stochastic process, and that's the Kolmogorov forward equation, sometimes called the master equation in this context. But it's basically a partial differential equation that describes how the probability of finding the system in any given state changes through time. So W here represents the probability of finding the system in state S, E, I, and R at time t, then that changes through time in a way that is very straightforwardly reflects this transition diagram. So in each case, we have for each of these different transitions, we have 2 terms: a probability flow into that state from the other state, and a probability efflux, a flow of probability away from that state. And so there's a pair of terms for every one of those transitions. You notice that there's 4 here. What about the sampling one is missing? And that's because those 2 terms actually exactly cancel each other under the assumption that a sample doesn't actually change the state. So the reason I'm showing you the Kolmogorov forward equation is, 1, it's cool, but 2, the filtering equation we're ultimately going to derive is going to resemble this. And so it's good to see what it looks like now. Okay, so I told you what I mean by a population process. Now I'm going to say that every population process uniquely induces a genealogy process. So we need to think a little bit about what a genealogy is. So of course, a genealogy is basically, a genealogy is a representation of the pattern of shared ancestry amongst a set of viruses. And if we're talking about viral phylodynamics, so and that is often represented using a kind of a tree structure. So in this diagram, the black dots here at time 10 represent lineages that are present in infected individuals. And the gray lines show the pattern of shared ancestry. So between, say, these 2 lineages, there was a common ancestor that was found at this particular time here. The blue dots show samples that may have been taken. So this is just a hypothetical example. And all of these lineages then coalesce as you move backward in time until some point in time where we agree to stop the whole thing. So we're just not interested in what happens before this time. So that's the tree representation of a genealogy. But of course, there's more going on here. So if we go back just a minute to this diagram here, you notice that I've shaded in 2 of the compartments. So each of these boxes is a compartment that contains, at any given point in time, contains some particular hosts, some number of hosts. But the hosts that are in the E and the I boxes are distinguished by the fact that they also contain a viral lineage. And because these individuals are distinct from those individuals, statistically speaking, but all the individuals within each of these boxes are statistically indistinguishable, we have to treat these 2 classes of viral lineages as distinct. So I'm going to call these demes. In this case, we have 2 demes, and I've shaded them here with gray. There's an E deme and an I deme. And so going back to this genealogy, we can note that each one of these lineages at each point in its history had to be in one or the other deme. So we can represent that, for example, using color. So if I've here indicated the E deme with the blue color and the I deme with the yellow color, then this would be a more complete picture of the history of those lineages, because it tells us not only where the shared ancestors are, but also where in the demes these individuals were. And I'll refer quite frequently to the coloring of a genealogy, as a shorthand for talking about this. Okay, so let's let this D here represent the set of demes. Here, there's the 2 demes in this case, but in the previous examples, we had more demes. It's also possible to have just one, but in general, we'll have a countable number of demes. And when we look at the Markov state transition diagram, we'll use the term U to refer to the set of all possible transitions. So here there are 5 possible transitions, and I've just used these labels to distinguish the 5 different possible transitions. So what I want to do now is show you how how the genealogy process is induced. So to do this, let's just focus on a very narrow strip of time, say an infinitesimally wide strip of time, and have a look at what happens at that time. So each of these little panels shows the lineages that cross this narrow strip of time, and we notice that there are, depending on what goes on in the population process, there are 5 distinct kinds of things that can happen in a genealogy. So, for example, if we have a transmission event, we have a situation where at the time of that transmission event, one lineage, in the I deme in this case, gives birth to a second lineage. So we have these birth type events that are possible. We have death type events that, well, they correspond to recovery events in the SEIR model. If an individual who's in the I category recovers, then that lineage terminates. It doesn't persist into the future. And in fact, it's lost from the genealogy. So it disappears. So I've signified that by putting the dashed line there. So immediately after this event occurs, this lineage is now erased. Oops. Oh, this mouse is very, very sensitive. Okay, so I'm sorry. We can have a migration type event. This, for example, the disease progression from the E category into the I category, a lineage moves from one deme to another. And so that's a migration type event. We can also have a sampling type event where an individual, say in the I deme, is sampled, but that sampling, according to the model, doesn't actually do anything to that lineage. And of course, it's also possible that there are events in the population process that have no effect on the genealogy. They're neutral as far as that goes. With these 5 basic types, and they coincidentally represent, each of the 5 types in the SEIR model is exactly one of these elementary types. But it's also possible to put these together into various kinds of compound types. So, for example, in the classical Moran model, that corresponds to the Kingman coalescent, you have events like this, where a lineage gives birth and simultaneously another lineage dies. So that's a compound event. That's a conjunction of these 2 things. You can have an event where sampling coincides with death. So, for example, if sampled individuals are quarantined, then they might effectively be removed from the transmitting population. And we can have, there's no reason why we have to limit ourselves to just single offspring events, and here's an example where a birth event involves more than one offspring. So we can combine these things in lots of different ways. But the point is that once we have the Markov transition diagram in this set of U's, for each different kind of event, for each different element of this U set, there's a particular event that occurs in the genealogy. In this way, a stochastic population process, let's call it X, of the S, E, I, and R in our example, induces a stochastic process G on the space of genealogies. This map from X to G is not a deterministic map; it's a random map, and that randomness is there because the identity of the individual host, of the individual viral lineages to whom those events occur, is a layer of randomness over and above that in the population process. The key assumption that makes everything that follows from here doable is the assumption that I kind of hinted at before, that the lineages within a deme are exchangeable one from another. In other words, that there is no more structure in the stochastic population model than is implied in the population process. So this is kind of a subtle point. It's often asserted, I've seen it asserted many times, that once you write down a compartmental model, it follows that all of the individuals in the box, each box, are identical to one another. They statistically indistinguishable from one another. In fact, that's actually an additional assumption that one has to make. But it's kind of an assumption. You can forgive people for not seeing that it's an additional assumption, because it's kind of completely in the spirit of the model, in the sense that it adds, it's the minimal assumption that adds no additional structure to the model. Okay, so I've shown you in a kind of a constructive fashion how one goes about deriving this genealogy process that just comes naturally from the population process. To give you a little bit better sense of what that looks like, let me show you some animations. So many of you will have been familiar with the, can you see this, Eric?
I can see this. I'm going to adjust things so it's a little bit clearer. Okay, I
Okay,
think that's fine. If you can make it a tiny bit bigger, that's
there
good.
we
But
go. Okay, so the Moran genealogy process is, as I said, is a sort of a fixture of classical population genetics. And so it may be familiar to many of you. So this is just a simulation of the Moran genealogy process based on that construction that I just showed for you. So you can see the black dots moving to the right as time progresses. The structure of the genealogy is changing itself dynamically through time. And in particular, you'll notice that the common ancestor of the whole group, of course, shifts as lineages die out. Then they're lost, and the structure of the genealogy changes. So it consists both of the addition of new structure at the leading edge and the loss of structure deeper in. If
so,
we
Aaron,
supplement,
can I just ask a quick question?
yeah,
This is very helpful. That's beautiful. One thing I'm trying to understand is when you are calculating things in a model like an SEIR model, we need these dots to be labeled by the type of event that is
we
there.
don't need them to be labeled. We can have them labeled if we want. Here, there's only one kind of event, so it doesn't happen. I'll show you in a minute a simulation from a more interesting model.
Okay, so you're saying that if we're considering a different type of model, the genealogy then would have labels, corresponding labels, like each dot would be labeled by what type of event was happening
Well,
on.
if we were simulating it, we could affix those labels.
Okay.
Right? There would be no cost to that. We would know those things. And so,
Mhm.
yes, we could label them. It doesn't follow what you said. I mean, you said we would need them.
Well, I guess I'm thinking about inference already.
Yes, you're getting a little ahead of
Okay,
ourselves. So what I'm going to
we'll get
show you
there.
is that this genealogy process, which follows as a consequence of the population model, needs to be projected down onto a smaller space where we lose a lot of that information in order to represent a realistic inference problem.
Mhm.
Like because I think where you were going is that we don't know those labels. We don't know that stuff. And so what ultimately we're going to do is see how we can nevertheless do inference despite the lack of that. But we're going to creep up on that. Do it in a couple
Wonderful. I
steps.
love it. Okay, let's stay in
Okay.
simulation land.
Okay. So yeah. So here we are in simulation land. So we know we potentially a lot more than we would in reality. But we'll get there. So here's what we've got: sampling that's going on through time simultaneously. So this just shows. So the blue dots show up when samples are taken. And so this is what the sampled Moran genealogy process looks like, where we have not only the Moran events with a simultaneous birth and death, but also sampling events. Now let's go back, press and hold. Let's go back and look at, as I said, a slightly more interesting process. Here's one where we have two demes. This is that SEIR. So we have the E deme and the I deme. And so here, Eric, you see I've got two different colors on the genealogy. I guess I've got gray and yellow this time instead of blue and yellow. But the different colors represent the demes in which those lineages are found. Again, the black dots represent the extant individuals, and the green ones represent events that occurred in the past. So we have events, for example, like this one that presumably was a migration event. as well as branching events represented. And you can see that as time goes on, of course, the epidemic burns itself out. And if we haven't taken any samples, we got nothing left at the end. That's a consequence of the fact that the death events or recovery events in this model erase the lineages behind them. But samples, of course, are not erased. That's the whole point of samples. And so if we sample that process, then we might have something like this. So we can see what the this is just one realization of a genealogy that's left after, well, here it's almost extinct, but not quite extinct, after the epidemic has passed. All right, so hopefully that gives you a little bit of a sense of what I'm talking about. The rules that I was showing you about how you go about deriving this basically are just the rules that you apply at each event that occurs in the population process. You manipulate the genealogy in a particular way, and that leads to what I just showed you in terms of the animations. Okay, so now let's get to Eric's question. So I've shown you how a population process induces a genealogy process. Spent a little bit of time talking about what that looks like. And now let's start to talk about how that relates to data. So this thing that we've just been simulating, we can call the full genealogy, right? It's got, not only does it have the relationships amongst the samples, but it's also got the relationships amongst the extant individuals, some of whom have never been sampled, right? So these are somehow in the mind of an omniscient being, but not in the data set. It also has the history in terms of the deme occupancy, right? In terms of where in the demes each lineage was at each point in time, including the exact moment at which the lineage moved from one deme to another, and so on. So this is emphatically got a lot more in it than we can see. So in terms of deriving doing the mathematical work, it's convenient to view the relationship between this and data as a two-step reduction or a two-step projection. So the first thing we can do is throw away all the portions of the genealogy that are unsampled, all the things that are not ancestral, all the portions of the tree that are not ancestral to any sample. And if we do that, so we just throw away the black dots and everything that goes back to the nearest blue dot, that gives us what I call the prune genealogy. So for fairly obvious reasons, right? So this is the prune genealogy that corresponds to that genealogy. And this is closer, but to what we might actually see. But of course, we still have the color, the painting of the genealogy. And so that is, we need to go one step further. But I'm going to do this, as I say, in two steps because it makes the mathematics easier, and I want you all to follow and understand. So let's look at the relationship between the full genealogy and the prune genealogy in terms of what happens to it. And so to do that, we can look again at an arbitrary but narrow strip in time, infinitesimally wide strip in time, and just focus in on the local structure of the prune genealogy. So in this diagram, it's the same kind of diagram I showed you before, but the upper row represents the unpruned genealogy, the full genealogy, as I was calling it. And then the corresponding one just underneath is what happens with a pruning, right? So a possible pruning. So here, suppose we've got at this little strip that we're focusing on, it contains a birth event. Well, when we prune, some of these lineages are going to be lost, and I've indicated them with the dashed line. So it may happen that this yellow lineage here was not ancestral to any sample, in which case that would be pruned away, but the blue one was, say, in this case. So we would have, at the end, a diagram that has 1, 2 yellow and 2 blue with what looks like a migration event here, right? A yellow to blue transition. So we would just simply see that. In this case, we have a true migration event that occurred in the full genealogy, but after the pruning, we would lose some of the other lineages. In this case, we've just lost some lineages that are unrelated to that event. And here's an example where we had two samples that were taken simultaneously in two different individuals at that time, and one of them, the descendant lineage gets pruned away, and in the other it doesn't. So we have, in this case, this would show up as a leaf in the prune genealogy, and this would show up as you might call an inline sample, a sample that's not a terminal leaf. So the point here is that the mapping from the full genealogy to the prune genealogy is a deterministic mapping, and one can carry that out. This is just showing you what that actually might look like. Okay, so we're going to need a few different quantities in order to be able to write down the equations that are the goal of the talk. So N will be the occupancy of deme I when the system is in state X. So we can get that by just looking at the top here. So the occupancy of the yellow deme is 1, 2, 3, 4. 4 yellow lineages in the population, and 4 blue. 1, 2, 3, 4 blue ones. So the occupancy would be 4, 4 in panel A, whereas in panel C there would be 2, 3 blues and 1, 2, 3, 4, 5 yellows, right? So the N I'll use to refer to the occupancy. We get that by just looking at the population process itself. We don't even need the genealogy for that. Okay. Now, for every event type, remember U is the different type of state transition. So for each event type, there's a production. And I want to use the symbol R to refer to the production of an event of type U in deme I. So that's the number of lineages that are to the right of the event, the number of lineages that are involved in the event that are descendant from the event. So in panel A, this is the event in question. There are 2 lineages that emerge from it, 1 yellow and 1 blue. The production is 1, 1. And this is, again, we're getting this strictly from looking at the top row here. In panel C, oops, whoa. Wow, I can't use the mouse wheel. In panel E, there are 2 lineages emerging from this event, and they're both yellow. So the production is 0, 2. Similarly, looking at the bottom row in the prune genealogy, the corresponding things are also important quantities. So the lineage count is the number of lineages that are actually present, right? So in panel B, there are 2 yellow and 2 blue. So lineage count is 2, 2. In panel F, there are 2 blue and 2 yellow. So again, the lineage count is 2, 2. And then finally, what I call the saturation is the number of unpruned lineages in deme I that are descendant from the event. So this is basically the thing that corresponds to the production, but in the prune genealogy. So here is the event. We just have 1 blue and 0 yellow descendant from that event. So the saturation is 1, 0. And here we have 1 yellow and no blues. So the saturation is 0, 1.
Okay, so this is okay. The number of unpruned lineages. So if it gets pruned, it does not count on the saturation. So
that's right. It counts in the production, but not in the saturation.
I mean, already I'm sort of thinking about the fact that the pruning sort of depends on a lot of downstream stuff. I mean, I'm sure we'll get to that.
It
Do you
does.
want to anticipate that at all?
It does. Well, what we're going to see is that there's there's the Markov process that gives us the full genealogies. There's also a different Markov process on the space of prune genealogies, and also ultimately when we throw away the color one there. And those are the things we're actually going to need to think about when we work out the probability distributions, which are what the final goal is.
Cool.
But we're creeping up on it. So obviously the saturation has to be less than the production, and you can't have more production than you have occupancy, and similarly you can't have more saturation than you have lineages to saturate, and those have to be smaller than that as well. Okay, so So we've got about 10 minutes left. I'm quite close to wrapping up, but I want to know how much, is it traditional to leave a little bit of time for questions at the end?
It is traditional.
Okay.
I
So
think,
let me,
everything has been super clear. I think you should just go for it and not disrupt. I think tomorrow.
okay. Well, I'll show you a few things and I'll go fairly quickly through this. And like I say, we have the whole next talk
Yeah.
To go into more depth.
Great.
So it turns out to be a little bit subtle how one actually mathematically represents this genealogy. We're used to making these trees, but a key thing that makes the mathematics work and makes the proofs possible is representing these genealogies as functions of time. But we'll talk about that next time. If we throw away the color, I call that the process of obscuring the genealogy. So we get something called an obscure genealogy, which has got the topological structure and the geometrical structure of the prune genealogy, but doesn't have the color. And we get that by just, that's just one function of time. Okay, so finally, we need one little bit of notation for the equation that's going to follow. And so I'm going to use this symbol here. This looks like a matrix. It's not a matrix. It's a combinatoric quantity. It's just a number. And it depends upon the four numbers that I defined, or four vectors of numbers that I defined before: the occupancy, the lineage count, the production, and the saturation. And it's just a combinatoric quantity, which is the product over all the demes of ratios of binomial coefficients. And so that's a number that lies between 0 and 1, and it satisfies this identity. And you'll see that it crops up. And so this is, we want to be able to write down, we want to be able to do this procedure for any given model. We need this kind of generality. All right. So the first of the theorems that I'll talk about in detail next time, but I'll just indicate here, and is what's the probability density function for a prune genealogy. So given a genealogy that looks like this, what's the probability that we got that under a given model? And so the way that theorem is going to look, and I'm suppressing some of the detail here, is, whoops, I can't. Okay, is that suppose we have some prune genealogy with a time depth t. I'm going to define this quantity phi here to be basically the product of a binomial ratio and an indicator function that's either 0 or 1, depending on whether the local structure of P is compatible with an event U at that time, right? So for example, if we're at a point in time when there were no samples taken, that's obvious from looking at the data, then if U is a sample type, well, that's going to have to be 0. And so Q is enforcing that, but that's basically what Q is doing. So anyway, so that's just a factor. And the theorem says that if W satisfies the initial condition that the model gives us, together with a filter equation of this form, so then the likelihood is just the integral over all states of the W. So let's look at this. I'm not going to go into detail on this because I haven't even defined some of these terms. This is a very general expression. In order to even understand it, I'd have to spend a bit of time on the notation here, and I'm going to do that in a month's time, around the 11th. But I'll just point out to you that you get an equation like this, and it's got two kinds of pieces. One, a continuous piece for events that are for times t that are not in corresponding to events in the genealogy, and one at the events that are in the genealogy. Okay, so let's, instead of doing the math and explaining exactly what that equation is, I'm going to postpone that. I'll just show you some examples. So let's take a simple SIR model here. And in this simple SIR model, the filter equation, the continuous portion looks like this. And so this looks, if you look at it, it resembles quite strongly the Kolmogorov forward equation for this model, but it's got some additional factors that are associated with, like this one, and there's another one in here that are associated with the binomial ratio. So this is one of those binomial ratios for this model and what it looks like. Okay, so and then there's the discrete portion or the singular portion of the filtering equation, which basically says if we're at a branch point we modify W by a multiplicative in a multiplicative way. If we're at an internal sample, an inline sample, we modify it this way. If we're at a terminal sample, we modify it this way. And so here, the tilde is just referring to the left limit, sort of the value of W immediately before the event, instantaneously before the event, and this is the value instantaneously after. So anyway, so those are the equations in this case, and one can apply those in a very simple way, in a very standard and straightforward way to infer, what's the probability of this genealogy. And here I've just calculated the probability of this genealogy as I vary one of the parameters, in this case, the loss of immunity parameter, over some range, and you can see the likelihood, or the log likelihood, has this characteristic shape that you expect it to have with the true value, because this is simulated data, in fact, the true value lying up here close to the top. That's a particularly easy case because there's only one deme. It turns out that both the coalescent likelihood, the Kingman coalescent likelihood, and the likelihood of the linear birth-death process, both are special cases. They both fall out of this formula in a natural way, and you can see that in the case of the linear birth-death process, just through this simple numerical calculation. Interestingly, to do this, the Moran case, the Kingman coalescent case, is quite straightforward to do analytically. This guy is still resisting my efforts to derive the analytical solution, but it sort of has to work, and the numerics bear it out. So, sorry. Next time, what I'm going to do is spend more time on the mathematical derivations to hopefully give you a sense of why this all works and what the equations. I'm going to also go further to actually deal with the problem that we're really interested in, which is what's the likelihood of an obscure genealogy when we don't have all of this extra information. And that adds an additional wrinkle to things. So when we have a genealogy that looks like this with no color in it, how do we go about deriving that likelihood? And I'll show you how that's done. So I'll both prove that first theorem, and I'll show you the next theorem, which writes down a filter equation for the obscure genealogy. So as I just briefly mentioned, this theory corrects in the sense that it shows some correction terms in those equations that are not present in the earlier methods of, for example, the Rasmussen and Volz. It also extends all of these, both the coalescent-based and the birth-death process-based approaches, in the sense that it gives the exact result, which in special cases reduces to the simpler forms. All the computations, and this is the really important thing, is that all the computations can be carried out forward in time, which means that in principle, any model, well, any countably structured Markov population process model can form the basis for an inference procedure, at least in principle. Whether one and how one can efficiently compute that is another question, and that's an important thing that I'd be very interested in talking with you about. But also importantly, there's a great deal of flexibility here. You can do anything from conditioning on the samples to modeling the samples. The sampling effort and modality can vary in an arbitrary fashion with time, or almost arbitrary fashion with time. And so this really helps, should help a lot in terms of being able to accommodate the kind of variety of scenarios that we're interested in, people are interested in. And because it's all forward in time, the standard approaches for assimilating time series data can be applied at the same time and in the same calculation. The details can be found in a archive preprint, and there's some references. So I just left a little bit of time for questions. I'm not sure what the rules are, but I'd be happy to take any questions if that's even possible.
Absolutely. Yes. Wonderful. Thank you very much. I mean, that was extremely clear, but I do encourage people to post comments. The first thing I want to do is just, if you don't mind, just to go back to the definition of W, just to make sure, because I know that's going to come up a lot.
This W, for example?
Well, for example, so can you just say explicitly, so I mean, in other frameworks, like W is something about like what is the probability density.
So this is a really interesting point, Eric, right? This is actually a really subtle and difficult point. And this is actually, in fact, the thing that I found hardest to wrap my head around in all of this. So in the Kolmogorov forward equation, W is a probability.
Mm-hmm.
At any given point in time, it's the probability density of finding the system in a particular state at that time. So that's straightforward. You can really intuitively, you have a natural intuitive understanding of what that means, right? Where by intuition, I mean, excluding people that don't know what the hell probability is. But if you can reason probabilistically, you get some kind of an intuitive notion of that. But this W is different, okay? This is actually not a probability. Emphatically not a probability. And what does that mean? It's not the probability of anything. So another way of saying it, the sort of more precise way of saying it, is that there is no event in the sigma algebra that corresponds to the population process of which this is the probability, right? Because this W contains elements that derive from events that have not yet happened.
Yeah.
So it's not the probability of anything. And if you try to think about it as the probability of something, you get into paradoxes of the usual sort that form the basis of science fiction movies, right? So you have events from the future that are changing probabilities now, and those are explicitly forbidden by the model that you're wanting to propose.
That's exactly where I'm going.
Unless you're a Hollywood director, right? You're not interested in those models.
Because we're going to be doing trimming or pruning and stuff like that downstream.
Right. And so what they are, so I told you what they're not. What they are is they are a partial probability. That is to say, if you multiply them out, they're a term or a factor in the probability. And so all we're doing here, and this is the key mathematical trick, is we've just refactored the probability calculation in such a way that we can do it sequentially in time. Because the full probability you have to multiply across time and across states. And here we're doing, you can do it either way. We're doing it across states, but within a time. And we end up with so the key thing is that, as the theorem says, is that it's only a probability at the final time. At all points before that, it's not a probability of anything.
Okay, so we're not taking each of these W's and multiplying them together. We're getting it all the way. Wait, no, but we are integrating. We're
integrating them through time. That's right. And they're effectively, and here there is a product going on at these discrete points in time.
So, okay, so if we want to calculate a likelihood, we have to solve a collection of these ODEs to get these W terms, and then take an integral.
Yes. And so in principle, this is, if the state space is infinite, as it often will be, then this is an infinite collection of ODEs. Okay, so you obviously need something clever to solve these equations. What I'm showing you here is just a sequential Monte Carlo calculation. So using a Monte Carlo solution, but in this case, it's the Monte Carlo error. That's where it's represented by these error bars. That's quite small. I can make that very small, just with not much difficulty with this small problem. And that's going to be, in a nutshell, that's the source. That's the computational challenge, and that's sort of the frontier where I'm at right now, is sort of cracking the nut of making this, going from a theory that tells you in principle what the likelihood is to a practical calculation in specific instances.
Cool. Well, speaking of which, Tanya.
Here's this question from Tanya Stock. Hello, Tanya. It's good to. I wish I could say see you, but I can at least talk to you. So let's see.
How does computational timescales models get more complex? In particular, how do you?
That's the question, right? And so obviously that's the fundamental question. And so the answer is that it's early days, right? So I've shown you only the theory right here. So I've shown you some results of some SMC calculations, sequential Monte Carlo calculation. Those are done in the most naive way possible, because that was just the first result. So those behave then in the naive sequential Monte Carlo. We know how those things scale, right? As the number of lineages gets large, then actually it's the number of, not the total number of lineages, but the width of this tree gets large. The number of particles you need to obtain a Monte Carlo estimate of a given with a given variance goes up exponentially. As the number of demes goes up, there's presumably an exponential relationship there as well. So the prospects for the naive approach are not great, right? On the other hand, there's nothing in the theorem that forbids us, sorry, where's the theorem? There's nothing in the theorem that forbids us, as we'll see, from doing more clever things that are equivalent to this. And when we look at the obscured genealogy, so this is a result of mathematical interest, not of any practical interest, because we don't have this information. You're going to need to tune in a month to see the obscured, the theorem that's really the important point. There's nothing in that theory that you'll see when I show you that result, that there's a good deal of flexibility there. And in particular, one can borrow information from the future to make the calculation go more quickly. But this is an active research.
Cool. Okay. Well, yeah, I'm looking forward to that next time. I guess 2 questions. So the first is there, and filtering is not something, I mean, I'm familiar with SMC, but you talked about the filter equation. Is there something I should be reading up on before next time?
Should be. I would say no. So in the sense that, there's a book.
Oh, that's okay.
There's a book that I could not find. So there are filtering equations. There's a history of these kinds of things that goes back to the 60s. In the context of things like Gaussian processes. None of that did I find particularly useful. And the general theory of Feynman Kautz calculations I did not find useful either. So as far as I can tell, this particular formulation of filter equation, it's not terribly complicated, but it's novel. And so they're actually pretty handy. And have, you can study them in their own right, and they've got some interesting properties that help you in devising equivalent algorithms. But I don't know of any reference on the subject. There's an appendix in the archive thing where I work out some of the properties of the filter equation. You can have a look at that. Okay. Yeah, I haven't found anybody else working with these things. They're kind of nifty, actually.
Sounds like maybe just we'll be fine waiting for next week and see.
Yeah. That's probably right.
Okay. Well, that's been really fun. Thank you very much, and I'm very much looking forward to next time.
Yep. All right. Thanks again. I'll see you then.
Thank you. Bye.
Bye-bye.